A very good morning to all. I, Dr. Vishakha Kulkarni, on behalf of MNR Educational Trust and MNR College of Pharmacy, welcome you all on national webinar on animal models for behavioral pharmacological studies by Dr. Jitendra Chimakurti, Professor and Principal, Vignan's Foundation for Science, Technology and Research, Guntur, Andhra Pradesh. Before moving towards the webinar, I would like to introduce MNR Educational Trust and MNR College of Pharmacy in brief to the resource person and all the participants. MNR Educational Trust has established in 1974 by respected Sri M.N. Raju sir and continuing its successful journey with dynamic Vice Chairman Sri Ravi Verma Manthina sir. MNR College of Pharmacy established in 2004 under the aegis of MNR Educational Trust and working under the leadership of Principal Dr. P. Alagarsamy, who is a well-known author in pharma field with 10 books and recognized as a top 2% scientist in the world by Stanford University. College is equipped with infrastructure to carry out day-to-day academic work and research facilities to carry out the research work. College is approved by AICTE, PCI and affiliated to Osmania University. With this brief introduction, I would like to request Principal Dr. P. Alagarsamy sir to give welcome address. Principal sir, over to you.
Okay, thank you, Dr. Vishakha. On behalf of the management, Sri M.N. Raju garu, Chairman MNR Educational Trust, and our Vice Chairman Sri Ravi Verma garu, and on behalf of all the faculty members, I welcome today's esteemed speaker, Dr. Jitendra, who is one of the well-versed in the pharmacology subject. So thank you for accepting our cordial invitation, sir. And I hope all the students will make use of this wonderful session. I welcome all the participants throughout the country. So this is one of the good session for those who are in the pharmacology loving peoples, because nowadays the pharmacological or the core subject seminars are very less, and he is one of the good eminent in this field. So let us make use of this webinar. And with these few words, I welcome again one and all the participants to this national webinar. So I request Professor Saravanan to introduce the today's speaker, Dr. Jitendra, to the speaker audience.
A very good morning to one and all. It's a great pleasure for me to introduce today's speaker for this webinar, and who is a very good research scientist, and my research collaborators, and is none other than Dr. Jitendra Chimakurti, founder, professor and principal, Faculty of Pharmacy, Vignan College of Pharmacy, Guntur, Andhra Pradesh. So, sir completed his B Pharmacy in 2002 in Vinayaka Mission College of Pharmacy, Salem, and in 2006 he completed his M Pharmacy in Pharmacology specialization in CL Bade Mehta College of Pharmacy, Chennai. And in 2012, sir completed his PhD in Pharmacology in Acharya Nagar University, Guntur. And sir is having collaboration with many national and international institutes, and his area of research interest is behavioral pharmacology, and in the topics only today he is going to deliver a talk with us. And sir is a recognized PhD guide in Vignan's Foundation for Science, Technology and Research, deemed to be University, Guntur. Sir is a life member of APTI, and he is a registered pharmacist in India. Sir is an associate fellow of Andhra Pradesh Science Academy, and sir is a member of Board of Studies and nominee of CPC SCA. Sir is the peer reviewer for many international and national journals, and sir is acted as a chairperson and subject experts in many national and international conferences. Sir is also a examiner and question paper setter for many reputed universities in India and abroad. And along with that, sir is having one patent in drug delivery of gabapentin, and sir is published more than 30 national and international publications in well-reputed journals. And sir is presented many papers in national and international conferences, and he attended many conferences, workshops and seminars, and he also delivered many guest lectures in various conferences, webinars and FTP. Sir guided more than 25 M Pharmacy students for the completion of M Pharmacy degree, and sir is guiding 5 PhD students currently. And In addition to that, SAR is also organized on many FDP workshop and conference which are funded by DST, AICTE. And SAR has started his career as a lecturer, and from that he held on a various position in Bapatla College of Pharmacy till after completion of his M Pharmacy in 2006. And SAR is having a vast 15 years, more than 15 years of teaching and research experience. And 2018 onwards, SAR taken as a founder principal in Faculty of Pharmacy, Vignan Foundation for Science, Technology and Research, Deemed University, Guntur, Andhra Pradesh. With this brief introduction, I wholeheartedly welcome Dr. Jitendra Chimmakurti on behalf of MNR College of Pharmacy, staff, students. Welcome, sir, and we are here to listen your words, sir.
Thank you, Dr. Saravanan, for introducing me. Shall I share my presentation?
Yes, sir. Now you can share, sir.
Okay. Is it visible?
Yes, sir. Is it visible, sir?
Okay. Good morning, all of you. I have been given the information that many of the students and faculty are from many pharmacy colleges. And this animal models of behavioral pharmacology nowadays, because of the new regulations, is becoming restricted only to post-graduation or doctoral thesis. And that is the reason why I have chosen to give an introduction about what is an animal model. First of all, I would like to thank Professor Alagarsamy sir and my colleague in my previous institution, Dr. Saravanan sir, for considering me as a resource person for this webinar series, which is being organized by them. I have been observing their activities, and it is a privilege for me to be a part of this webinar series. And definitely, I hope I will be fulfilling their intentions of conducting this webinar. First of all, unlike other disorders, behavioral disorders are somewhat complex to understand because we have to mimic the behavior of humans in that of animals. In the case of other disorders, for example, if you take any ulcer, we will be inducing an ulcer with some inducing agent, and it is visible whether the ulcer is induced or not, and we can compare to what extent the drugs have treated that ulcer. Whereas in the case of behavioral disorder, we have to induce a disorder in the animal which mimics the disorder in the humans. And it is highly complex to observe such sort of things. And apart from this, there are multiple types of behavioral disorders. So starting from cognitive disorders to personality disorders or mood disorders, emotional disorders, adjustment disorders, there are different types of disorders. And it is highly difficult to induce such sort of variations in the behavior in the animal, which requires many factors. Fortunately, we are having a long list of such type of animal models that are available, which are clearly demarcated for individual types of disorders. But the problem comes is, what is the type of animal model that we have to select for screening any type of any type of behavioral disorder? And this depends based upon the type of drug, a type of inducing agent which we are using to compare in that study. So to be brief in this thing, so first of all, I want to introduce all to what are the different types of behavioral disorders. Generally, we know that personality disorders, under which we have schizophrenia, schizotypal behaviors, paranoia, and all. Whereas mood disorders, we have mania, depression, attention deficit hyperactivity disorders. Whereas under emotional disorder, we have anxiety, phobia, panic states, obsessive compulsive disorder, and post-traumatic stress disorder, of which minor type of behavioral disorders were adjustment disorders. Many of us know that there are most commonly known disorders for depression or anxiety and all. And I am not going into detail about all these type of common disorders, because though induction of a behavioral change in the animal is difficult, understanding or conduct of animal models pertaining to behavioral studies, it is very easy. It involves a very simple apparatus, and it involves only observation in most of the cases of a few parameters. There is nothing complex in performing any animal models pertaining to behavioral studies, and that is the reason why I am not going into detail about what are the procedures for conducting all these things, because these are simple. With a single reading, you can understand uh, what is the procedure that has to be followed in all these cases of animal models in behavior disorder. So, for example, you can see these are the animal models of depression. One is swim test, tail suspension test, learned helplessness test, neuricide behavior, which is a behavior of rats killing mice because of their aggressiveness, uh, antagonism of catalepsy, caution, stress model, and isolation induced hyperactivity. So these are the few common models, animal models of depression. In the same way, all of us know what are the animal models of anxiety: elevated plasma stress, food shock induced freezing behavior, dark light model, water maze test, staircase test, marble burying behavior. All these are different types of animal models, which are very easy to understand, very easy to perform, and which give very highly uh, accurate results, or we can say differentiable results, where we can clearly differentiate the observations in control animals or in drug treated animals. And in most of the cases, the inducing agent will be the environment itself. There is no some other chemical which has to be used to induce these type of behaviors. Even the environment, the type of equipment which we use itself will be acting as an inducing agent to induce such sort of behavioral disorder. Generally, as we have seen that behavioral models are generally very simple, easy to understand, easy to perform, which does not require any sophisticated equipment and all. I am here concentrating more on selection of the appropriate model for your studies. As we have seen, there is a long list of animal models for behavioral studies, such as anxiety, depression, or phobias, or schizophrenia, and all. We should be very conscious in selecting the type of animal model, though they are categorized under that animal models of depression or animal models of anxiety and all. It is very essential for us to be very specific in selection of the animal model, because animal model might be representing some behavioral disorder, but it is not essential that that animal model will satisfy all the required criteria for it to be an animal model for that disease, and for us to be confirming our observations pertaining to the therapeutic efficacy of our drug. So it is essential for an animal model to meet certain criteria, or it should have certain requirements. So we should be very specific in selection of this animal model for performing our screening. That is one thing. So at the end of this talk, you should be knowing what is the animal model that you should select based upon different criteria to assess the efficacy of your therapeutic agent. So the basic criteria which an animal model should fulfill is, one is analogy of symptoms. Analogy of symptoms in the sense, the symptoms that are observed in the human model or human diseased condition should be analogous or should be similar in the case of animals also. That is analogy of symptoms. But in some of the cases, we won't be observing such sort of similarity. For example, if you take in the case of screening any anti-diabetic agents, where if at all you have used mice as an animal model, mice, when you induce any hypoglycemia in mice, it causes convulsions. Such sort of hypoglycemic convulsions are not seen in the case of humans, whereas they are observed in the case of mice, where there is no analogy of symptoms. Though the treatment, if at all the anti-diabetic drug, if it is preventing such hypoglycemic convulsions, we are considering it to be having a therapeutic efficacy, whereas there is no analogy of symptoms. And one such criteria is there should be analogy of symptoms. This is necessary because to correlate or match that animal model which we have selected to 100% that it resembles the human model. So that is analogy of symptoms both in humans and in animals, animal model that has been chosen for this study. Other thing is, there should be observable and measurable behavioral changes in the animal model. It is not like very slight or minute behavioral changes which we should be depending upon. There should be a clearly demarcated variation in the control animal and in the diseased model. So that is another criteria which an animal model should fulfill. The behavioral changes which are observed in the animal should be clearly differentiable and should be measurable. So this is the another criteria that necessitates the requirement of an animal model. And there should be an inter-observer agreement. Means it is not like if I observe something and if I feel that this is valid, it will become an acceptable animal model. There should be a consensus that all the people should be considering it to be an ideal animal model for that type of behavioral disorder. So that is inter-observer agreement, or which we can say, which is widely accepted among the different people without any objections. And in the same way, the response to a treatment should be similar in the case of both the animal model, which resembles to that of in the human model. There should not be any variation in that particular thing. And another important requirement is reproducibility. So it is not like this animal, this model, it is observed only in this particular in few cases, not in all the cases. Such sort of exemptions should not be there in that particular animal model which is selected or which we consider it to be an ideal animal model. In the same way, the last thing is biomarker responses. This is somewhat related to the pathological states which are causing that disease or disorder in the animal model should be similar to that of the disorder in the case of humans. Means that factor which leads to the disorder should be common in both the animals and also in the humans. So these are the basic requirements for an animal model to be considered to be valid, or we can say ideal for our study. Analogy of symptoms, existence of observable and measurable behavioral changes, inter-observer agreement, same response to treatments in both animals and humans, reproducibility of the systems, and biomarkers. Taking all these things into consideration, many researchers have considered that validity of an animal model for any study can be differentiated into 5 major criteria. That is homological validity, pathogenic validity, mechanistic validity, phase validity, and predictive validity. Means if at all I am having a drug which is considered to be having an anti-anxiety activity or an antidepressant activity, I will search for an animal model, and my search should be in such a way that that animal model should be able to fulfill all these validity, all these criteria. Then I can consider that particular animal model for my study, and then I can consider that results which I got from this study are reliable and which are applicable in the humans also. So today we'll be discussing about all such sort of validities of an animal model. So with this we will be knowing whether that animal model which we have chosen is valid or not for our study, or to come to a conclusion that our treatment is effective in multiple dimensions or in multiple conditions. And this 5 different validities can once again be subgrouped based upon the criteria. So we have seen how homological validity, pathogenic validity, mechanistic validity, phase validity, and predictive validity are 5 essential criteria which an animal model should fill. Under that, we'll be seeing in detail about what are the sub criteria which that particular validity will be concerned. For example, homological validity means it is concerned with species validity and also strain validity. So species validity in the sense whether rat or a mice or a guinea pig or a rabbit or a monkey, whatever it is, whether that particular species is valid to be considered to study that particular behavioral disorder. That is species validity. So by following all these 5 different validities, we will be coming to a conclusion that if at all any animal model, if at all if it satisfies all these criteria, we can assure that the results which are observed in that particular screening study can be 100% correlated with that of the results in that of humans, or we can say if it is shown to be very effective in the case of animals, then automatically we can say that it will be effective in the case of humans without any variation. So starting from species validity till this treatment validity, all these things will be covered in this different validating mechanisms. In species validity, we have seen that whether that particular selected species is suitable to this particular behavioral disorder or not. For example, if at all we see that depression occurs in humans, which is commonly known, it might be because of some early environmental factors, or it might be due to some triggering factors which might make him susceptible to that disease. We should also be knowing whether that animal model which we have selected or that species which we have selected is also having the same parameters, means whether it is also having such triggering factors, whether it is also having such early environmental factors which might lead to that particular depression. So that is species validity. The same way, strain validity. And in the species itself, for example, in the rat itself, there are different types of strains. Our study should also be like that, whether that particular identified strain of that animal, whether rat, Vistar or Sprague-Dawley or Nude or Pulp-C, all such, whether that is suitable for that study or not. It is like some strains are resistant to some sort of activities. They might be having some abnormality in their genetic makeup, which might not contribute to the occurrence of the disease, or which might not contribute to the occurrence of the facilitating the treatment with the drugs. So, in this way, we should be specific, starting from its identification of the animal, identification of the strain of the animal, till the treatment biomarker changes. All these things are to be validated before selecting an animal. So this is an example. We'll be seeing about all the other things as we go on. So before going on to this validity, we should understand how we will be differentiating a disease model. So generally, we can differentiate a healthy animal or a healthy human to a diseased condition in 3 stages. The first stage is, once again, it is a healthy individual or a healthy organism, whereas the second stage is susceptible to a disease, and the third stage is treatment of that particular disease. So these are the 3 different stages in any disease condition. For example, an initial healthy organism might directly be susceptible to a disease because of various factors, because of some inducing factors, or because of some pathogens, or because of some disformation, or we can say damage to any tissue, damage to non-functional system. All these things will lead to occurrence of a disease. But in some cases, there might be some epigenetical changes in that individual, making that particular individual more susceptible to that disease than others. And this can be differentiated in stage 2, that these are the early environmental factors. Early environmental factors are that particular factors which might be making that individual more susceptible, or we can say vulnerable to that particular disease. And he will be having that disease only if he is triggered for that vulnerability. Means it is, we can say, he is the possible individual who will be susceptible to a disease, provided he should be exposed to that triggering factors, which will be making him making the symptoms, or we can say diseased. Whereas in some cases, the triggering factors directly will be converting an initial healthy organism into a diseased organism. Whereas the third stage is treating with a therapeutic agent, and treating with a therapeutic agent, once again, making this diseased individual back into a healthy individual, or diseased individual back into a vulnerable individual. And this is the third step. In this way, a diseased model can be understood. And our selection of an animal model based upon the validities can once again be correlated to different steps of this diseased model, which we'll be seeing later. First, we'll start with this homological validity. In homological validity, we have seen that there are two different validities which we have to take into consideration. One is species validity and the other thing is strain validity. And here it is essential that the behavior in that animal model which we have selected and the symptoms of the disorder that are observed in the humans or in the animal, they should be homologous, or we can say they should be similar. It means the animal model in which we have selected should produce the same symptoms as they are observed in the case of humans. Unlike in the previous case where we have seen anti-diabetic activity in the case of mice, where preventing from occurrence of convulsions is considered to be its anti-diabetic potential, though such convulsions are not seen in the case of humans. And this is one thing which is an example of this particular similarity in the case of symptoms and similarity in the case of behavior. So since we are dealing with behavioral disorders, so this animal model should be having that particular similarity. In the case of symptoms, it is showing with that of symptoms in the human. And this validity will also see whether that particular test which we have chosen, that particular animal model, is accurately measuring what it is intended to measure. That is another criteria which homological validity will assure. Then we can say it is homologically valid. And based on this, we should be selecting that particular species. And based on this, we should be selecting that particular strain. It means that behavioral abnormality should be there in that strain of that species, and the symptoms should be observed in that particular strain of that particular species, and it should be accurately measuring that particular change in the symptoms after the treatment. So that is regarding homological validity. So this is the first thing, means there should be homologous acceptance between the humans and between the animals which we have selected for the study in terms of symptoms and in terms of the behavior. You can see here when we have overlapped these particular validities and sub-validities in the diseased model. For example, here you can see this homological validity or species and strain validity will be in the initial cases where that animal is having all the characteristics or is having that potential to produce that symptoms similar to that of the symptoms in humans in any selected disorder. So that is homological validity. So here you can see this is the homological validity where the selection of the species, selection of the strain will be validated here. So once we have selected the species and once we have selected the strain in the species and we have validated that this is the suitable or this is the appropriate animal, appropriate strain of the animal, then we have to move on to other types of validity, means other types of conditions, whether it is fulfilling it or not. Next is the pathogenic validity. So here in this type of validity, it sees about the similarity of the factors that are leading to the occurrence of the disease in the animal model with that of humans. So in the previous case, it is only the symptoms and the behavioral changes. Whereas in this case, it is concerned with the factors that are inducing that particular disease in the animals. They should resemble to the factors that are inducing the disease in the human. So that is pathogenic validity. Means the pathological changes which are occurring in the animals and which are occurring in the humans should also be similar. So if it is similar, then we can say this animal model is having pathogenic validity. In the previous case, if the symptoms are similar and if the end behavior is similar, then we can say that that animal model is having homological validity. Whereas if the cause of that particular disease is similar in both animals and humans, then we will say that it is having pathogenic validity. And this pathogenic validity can once again be subdivided as ontopathogenic validity and triggering validity. So because in this model, we have seen that there are two different factors which are involved in conversion of an healthy organism into a diseased organism, or a healthy human into a diseased human. So first factor is early environmental factor, and the other thing is triggering factor. So an early environmental factor will not be directly making any animal into a diseased animal. It is only making the animal more vulnerable to the disease. That animal will remain in that vulnerable state unless it has been triggered by some other factors. That triggering factors will be converting this vulnerable animal into pathological animal, or we can say diseased animal. So hence, this we have seen that pathogenic validity is nothing but it is the factors which are involved in converting a healthy individual into a diseased individual are to be similar in both animals and humans. So hence, it is related here in these cases where ontopathogenic validity is nothing but it can be considered as the early environmental factors which will make an animal into a vulnerable animal, whereas triggering validity is nothing but validity of that particular triggering factor which converts a vulnerable animal into a pathogenic animal. And a triggering factor can also directly convert an healthy animal into a diseased animal. So that is regarding the pathogenic validity in a diseased mode. So we have seen ontopathogenic validity is the validity of the early environmental factors which are making the animal vulnerable. The resemblance in the animals and in the humans, it has to be validated. If there is no resemblance, then we can say that this particular model is not satisfying ontopathogenic validity. In the same way, the triggering validity, that particular causative factor which is making a healthy individual to a diseased individual is called as triggering validity. We can say that factor which is causing a disease in the animal is similar to the factor that is causing a disease in humans is same, then we can say this model is accepting the triggering validity. So for example, in the case of animal model of depression, where maternal care deprivation, in this case, the newborn litter will be separated from their mother rats for about 3 hours a day from day 1 to day 40. So immediately after the birth. So this is a model which is called as maternal care deprivation. And here, this particular separation from its mother will make that particular animal to be more vulnerable to depression in the adulthood. And hence this particular factor, it can be considered as a early environmental factor which is making that animal vulnerable. And the triggering factors are factors, for example, after in the adulthood, once again, such any other factors which lead to depression. If that person is encountering to such isolation or any other separation, there is an increased chance of him getting that particular depression, or because he is already vulnerable. The same thing can also be observed in humans, because these are the factors which the thing which is occurring in the case of rat says there is an alteration in the hypothalamus pituitary axis, HPA axis alteration it occurs. The same thing is observed in the case of humans. Hence, we can say that this particular model, it is accepting, or we can say it is fulfilling the ontopathogenic validity, and hence it can be used in such sort of disorder. Triggering validity is nothing but in the adulthood, any other condition which triggers occurrence of the disease. Vulnerable individual is not a diseased individual. Vulnerable individual is having a potential to be converted into a diseased individual. And here, this is an early environmental factor, whereas the triggering factor later which occurs, such validity also has to be accepted in this case. Means ontopathogenic validity, the other thing is triggering validity. So this is the second type of validity. Then coming to this third type of validity, that is mechanistic validity. In the previous cases, before these validities were differentiated in this way, in the previous cases, there used to be only two different types of validities which all the researchers will be concerned with. One is external validity and the other is internal validity. So, for example, external validity is just applicability of the results that they observed in the case of animals to that of results that they observed in the case of humans. No other things will be considered, whether that species is suitable, whether any other species will be more suitable. All such sort of variations will not be seen. They will be simply testing. If results are shown, then they will interpret the same thing in the humans. That is external validity. In the case of internal validity, many other factors will be considered, are considered previously. That is reproducibility. Reproducibility in the sense whether this animal model, if it is repeated, whether it is producing the same results or not. That is reproducibility. And other factors such as inter-observer agreement, all these things also will be considered as internal validity. Biomarker validity, all these things are considered there. But later on, these are subgrouped in such a way that each and every factor will be covered before the selection of that particular animal. For example, we have seen homological validity and pathogenic validity, and the third validity is mechanistic validity. So here, this will be assessing the similarity of the mechanisms leading to the disease in the animal model and in the human disease. Whereas in the previous case, we have seen that in ontopathogenic and triggering validity, where the factors which are involved in inducing the disease are similar or not. Whereas here, in mechanistic validity, such resemblance between the animal model and the human model in the mechanism in which those factors are altered. So such resemblance will be observed in the case of mechanistic validity. In pathogenic validity, the factors, triggering factors, early environmental factors are, whether they are similar or not, will be valid. Whereas in this case, in what way they are changing, to what extent they are changing, all these things will be observed in the case of mechanistic validity. So the mechanism that is involved in the the occurrence of the symptoms. That is considered here. In the same way, the changes in the biomarkers or biological factors which are leading to that particular disease are taken into consideration. In the same way, the treatment with the therapeutic agents, the mechanism in which they alter this biomarkers or alter these factors are similar, whether they are similar in the case of both animals and humans. All such sort of factors are validated under mechanistic validity here. And for example, in few cases, the result of one mechanism interacting with other mechanism also can be occurring, where there will be an overlapping between these mechanisms. So such sort of things also will be considered in the case of mechanistic validity. So mostly it is pertaining to the mechanism in which the disease is occurring, involving all the factors which are similar in both humans and animals. That is mechanistic validity. Another important validity, face validity. And this face validity and another validity which we'll be seeing later, that is predictive validity. Both these things are widely accepted by many researchers. So this face validity is the one which validates the extent of similarity between the model and the disorder. Starting from the symptoms, starting from the signs which are observed in humans. The extent of similarity. In the previous cases, we have seen whether the symptoms are there, whether the biomarkers are there, whether the factors are there, early environmental factors or triggering factors or mechanism, whether they are similar or not, are being validated in all the previous cases. Whereas in this case, it is validating the extent of similarity. Means it is like we have seen that HPA axis is involved in that abnormality of maternal care deprivation, and we are also seen that the same thing also happens in the case of humans. And we are validating that as ontopathogenic validity is satisfactory, that in both the cases the same early environmental factor is involved, or the same triggering factor is involved. But the extent of similarity in both the humans and animals will be validated with this face validity. So face validity is the one which will validate the extent of similarity between the animal model and the human model. So this once again can be differentiated into ethological validity and biomarker validity. So for example, you can see this diagram where what is this ethological validity. So this is ethological validity, you can see here, which resembles the behavioral symptoms, and the biomarker validity, which resembles the biomarker changes. These are the two things which generally are altered in any diseased condition. So diseased condition means the biomarkers will be altered, neurotransmitters will be altered, or any enzymes, whatever it is. These are considered as the biomarkers because we will be estimating such particular neurotransmitters or enzymes to assess the degree of treatment or the degree of disease. So we have seen that face validity will validate the extent of similarity. When we have seen that if any one particular biomarker which is inducing any disease in animals is similar in humans, and the extent to which that it is inducing the disorder in animals is same or it is reflected in the humans, then we can say that it is having biomarker validity. And in the same way, if the symptoms that are shown in the animal with any abnormal change in the biomarker is similar to that of the symptoms which are shown in the humans, it means behavioral changes which we are observing. So if both these things are validated, means if they are more and more similar between the animals and humans, then we can say that animal model is fulfilling this criteria for face validity. So under face validity, we have seen one, it is ethological validity, and the other is biomarker validity. So for example, in ethological validity, where we are observing the behavior or outcome of that particular disease. Outcome of a disease is nothing but what is the symptom that we are seeing. And this symptom, it may not be the same symptom which is visible to us. It can be the same behavior of that particular animal or of the human. For example, the nest building activity of a rodent can be equated to the daily regular care that any human will take for himself. Daily activities of humans, they can be equated here. So it is not like the same behavior can be seen, but animals will be having their own behavior, humans will be having their own behavior, and that correlation, what type of behavior can be correlated to that of humans, because it is not possible to reflect the same behavior of humans in that of animals because of differences in terms of the intelligence or intelligence levels and understanding levels or interpreting levels and all, they are different. So the behavioral changes will not be similar, or we can say cannot be 100% reflected here. We should be considering that particular model, that particular behavior, which is similar to that of animals, which can be compared to that of humans. So that is ethological validity. Whereas in biomarker validity, that particular factor which is inducing the disease, or that particular factor which is altered the treatment, both these things are to be similar, and the extent of similarity also should be high. So then we can say that this animal model is satisfying the biomarker validity, so that it is clearing the face validity, providing if it is also having that ethological value. So for example, you can see in some animal models such as unpredictable chronic mild stress. So this is an animal model for depression. So here, in this case, antidepressants are effective only on chronic administration in the animal. The same thing should be observed in the case of humans also. It is not like they are effective on chronic administration in the case of animals, but they are effective on acute administration in the case of humans. Then we cannot validate both. If it is showing therapeutic activity only on chronic administration in animals, it should also show the same therapeutic efficacy on chronic administration in the case of humans. That is face validity here. And the last type of validity is predictive validity. So predictive validity is something which predicts. So here there is a correlation which is taken with the treatment outcomes, but not with the top pathological resemblance or mechanism of action of that particular. Means it is different from the previous types of validity. So for example, in the case of face validity, we have seen that the similarity of the extent of similarity of the factor that is inducing a disease, or extent of similarity of a factor that is treating a disease should be high. Whereas such sort of thing is not taken here. Only the therapeutic outcome is taken into consideration. It is not considering about what is the factor that is inducing the disease, what is the extent of similarity that is between the disease, is not considered in this predictive validity. Only a correlation is seen with the treatment outcomes. So here in predictive validity, so generally there is a similarity in relation between the triggering factors and the occurrence of the disease is taken, and a similarity between the therapeutic agent and the disease treatment is taken. Only the outcomes will be seen. What happens if this particular thing is occurring in the animals or humans? What will be the outcome of this particular change? Whether that outcome can be compared to that of humans in that of animals. That is predictive validity. So here it will be identifying whether the treatment with pharmacologically different drugs can be applied to that particular animal or not. If we are categorizing any class of drug as antidepressant, it should be able to treat different types of depression. That then we can say that this particular animal model is having predictive validity, and we should not say this will be valid only in this sort of depression and this sort of behavior and this sort of behavior only if the chemical biomarker is of this extent. Such sort of variations cannot be applicable to make that particular animal model to have predictive validity. So that limitation should not be there, and it should be identified that this particular treatment, it is animal model, it is suitable for studying pharmacologically different types of drugs, irrespective of the mechanism in which it is acting or irrespective of the biomarker it is altering here. So because it is only checking about the therapeutic outcome and not with any other pathological resemblance or mechanism of action. Apart from this, this model also will validate the potency of the drug, drug's response in the animals. So some drugs will be highly potent. So we'll be differentiating these drugs as to be more effective, less effective, or moderately effective. So such sort of variations also should be shown in the case of that animal. If such variations are also shown in the animal model, then we will be saying that this is satisfying the predictive value, because it is concerned only with that of therapeutic outcome. And predictive validity can once again be differentiated into induction validity and remission validity. So for example, induction validity is nothing but that inducing factor or the triggering factor which is triggering the disease or which is triggering the changes in the biological markers or biological factors to induce that particular disease. That is induction validity, means the same triggering factors are inducing the same disease to the same extent in the case of animals and humans. Then if it is similar, then we can say that it is having induction validity, means it is inducing the disease. This is the factor that is inducing the disease in this animal model and also in the human model. The same way, remission validity. So remission validity is pertaining to the treatment. Induction validity is pertaining to the induction of the disease, whereas remission validity is pertaining to the treatment. Whether that particular treatment is altering that biomarker changes or whether that particular treatment is giving the required therapeutic outcome. What is the extent of similarity in terms of animal model and in terms of human? So here that is pertaining to the treatment. So such validity in terms of inducing factors, validity in terms of treatment outcome will be covered by this predictive validity. So for example, I think we have completed the different types of validity. So for you to understand, there are 5 different types of validity, which are once again subgrouped. So total making them to about 9 different types of validity. So of which the first one is homological validity. So where species and strain validity is seen, whereas the next validity, it is pathogenic validity, where the ontopathogenic conditions and also the triggering conditions, which make that animal, or what is the pathological basis of that occurrence of the disease. That is pathogenic validity. In the same way, mechanistic validity is involved in mechanism in which the disease is occurring in animals and in humans. Whereas phase validity is biomarker validity and ethological validity. Predictive validity is induction validity and remission validity. So all these are different types of validities which an animal model should fulfill to make it more suitable for that particular study which we have chosen. So next time, whenever we are screening any anti-anxiety drug or anti-depressant drug or any other drug for the treatment of any behavioral disorder, we should take into consideration of all these factors based upon the previous research. So we should also include these factors to in our literature review to make sure that whether we are selecting the appropriate animal model. Because at the end of the study, once again, if you found that this is not the suitable animal model for your screening, this automatically will make your study less useful. So hence it is essential for all of us to make sure that the identified animal model, though it might appear, for example, elevated plasmase, it is a very simple model. Though it might appear to be a very simple model, unless it validates all the factors, making it more similar to that of a human disease model, it will be of no use performing that animal model. So here I have given two examples of two animal models uh, which are categorized, or we can say which are differentiated based upon this validity parameter. So one such model it is a model of anxiety, that is elevated plasmase, which we know. So for example, these are the 5 different validities, and these are the subtypes of validities, and this is whether that particular animal model is accepting that, is having that similarity between that of animal model and human model. So that is to what extent it is accepted. So for here you can see in the case of homological validity, the species validities, strain validity. So here the species, whether which we have chosen, it is having an ability to be anxious or not. So that is the first thing. If it is having that ability to get anxious, then we can say yes, it is accepting. And that particular strain of a species, there are different strains in a rat. Which particular strain is more suitable for this particular strain? So here these are a different strain which are categorized as high anxiety behavior rats. They are categorized as that particular strain, and this sort of strain has to be selected if at all we are performing this particular elevated plasmase. There are other strains such as low anxiety behavior strain, which automatically will not be producing the same anxiety. This is something like selection of animals in a rotorod apparatus. So for selecting an animal on a rotorod apparatus, initially we should place the mice on that rotating rod, and we should see whether that animal is able to walk or be holding that rod for a duration of about 5 minutes. And we will be including only such animals which could be able to stand on that particular rotating rod for our study. The same thing, if the animal itself is not having an ability to stand on that rotating rod, we cannot differentiate the observed results, whether the results are because of our drug or whether the results are because of that animal's behavior. So that is the strain validity. So here in this case, this particular strain will be more suitable for elevated plasmase. In the same way, ontopathogenic validity and triggering validity. Ontopathogenic validity are early environmental factors, which generally in any new place you will be having some anxiety, In the same way, the rat also will be having the same anxiety. So that is ontopathogenic validity. In the same way, triggering validity is, for example, if you hear when you are in a jungle, and as soon as you hear a lion's roar, that is a triggering value. In the same way, rat, when it hears some it's any other abnormal sound, automatically it also can become more anxious. That is triggering value. So that also it is accepted. Mechanistic validity is nothing but what is the mechanism that occurs in inducing that anxiety in animals and humans. That is accepted. In the same way, in this way, all these validities which we have seen previously are accepted in this particular animal model of rat in this elevated plasmid. So, hence that is the reason why elevated plasmid has become the most suitable model or most used model for anxiety. So, whoever is doing anxiety, the first thing which comes into their mind is elevated plasmid because of this individual validity. And the choice of elevated plasmid as a best model of anxiety has come into existence before all these particular things were identified or studied upon, because the extent of similarity, or we can say the face validity, is more accepted. Extent of similarity in the factors which are inducing the disease, extent of similarity in the response with the treatment, these are high. That is the reason why it has become an ideal model. In the same way, another anxiety model, which is called as novelty suppressed feeding. This also is an anxiety model for anxiety. And here you can see to what extent it is satisfying the criteria of this validity when compared to previous model. Previous model we have seen it is accepting all the criteria, whereas in this model it accepts only few validities, whereas it is not accepting the other validities. So, for example, this novelty suppressed feeding is, it is observed that rats, whenever they are in some new environment, or whenever they have come across with some new food, automatically their feeding will be suppressed. Means they won't be eating that food, or they won't be eating in that novel environment, or they won't be eating that new type of food. So that particular thing. Whereas it is not similar in the case of humans. For example, if we are going to some other new place, our feeding habit is not suppressed. In the same way, if we see something new, our interest in eating that particular thing will be increased. So this is entirely contradicting in the case of rats and in the case of humans. So hence, in such cases, it is not validating because there is no similarity in that particular thing. Hence, such type of animal model, it is not a suitable animal model for all types of anxiety. Only we can say whether this drug is having some anxiety, anti-anxiety effect or not, but we could not validate that particular thing, or we could not correlate the results that are observed in that particular animal model to that of human model. So in this way, it is essential to make sure before the selection of an animal model to what extent it is validating the results, or we can say validating the animal model, so that the results which are observed in that particular study can be interpreted to that of humans. So with this, we will be ending this today's talk, and once again, thank you for your patient listening, and over to Dr. Saravanan.
Thank you. Thank you very much, sir, for your detailed description upon the animal models and basically the validity concept. Thank you very much. There are questions, some questions are there, sir. I will read for you, sir.
Okay.
Do we have any animal model to depict the genotype for specific targets or for transporter proteins? Where can we access them? Can I read it again for you, sir?
Yeah, I could see the same question.
Yes, sir.
Genotypes for specific targets, definitely these animal models are there with specific genotypes, with altered genotypes for various animal for targets. But the thing is, they are not available for all types of disorders, or we can say all types of conditions. Only very few conditions where in the case of most commonly seen, where in the case of cancer, in the case of diabetes, or in the case of hypertension. Only in such cases we can have such sort of this thing, altered genotypes, so that we can direct our treatment to that particular target. And regarding transporter proteins, yes, all these things also are available, but the thing is they are not available here, and it is possible. Whatever is required by them, it is possible, because by knockout mechanism, the required gene, it can be removed, and that particular state of that animal model can be induced.
Oh. I hope the madam got the answer. Anyone is having question, we can unmute. Please ask, raise the hand. We can unmute the participants if anyone is having question. Sir, till that time, being a BPharm students, some BPharm students also are here. So some students are asking that whenever we are working on the animal models, is it mandatory to validate animal model, like going for the validity, or we can directly use the well-known model? We can go directly to work on this known models.
Well-known models are, that is the thing. Here I have shown an example of an elevated plasmid model. Before it is being categorized or validated under different criteria, it has become a most accepted model. It means the results that are observed with the treatment or the behavior that is observed in that particular animal, it is widely variant, or we can say it is clearly there is an observable difference which can be measured to a wide extent. So that is the reason why it has become a well-known animal model. So when coming to selection, it is definitely a well-known models can be selected, but it doesn't take much time to make sure that whether that particular animal model is satisfying all the criteria or not. Only with a brief literature review, we can see whether it is satisfying this homologous validity or pathogenicity that is involved, mechanism that is involved, or base validity, predictive validity. All these things can be checked. Once if we found that it is not validating any of the parameter, then we can add some other animal model which validates that particular thing also. So we'll be using 2 animal models so that we can complete the list.
Okay, sir. Okay, thank you, sir. Similar type of question is there. How a good model is identified, on what basis? So you answered the same way. I think this question is answered already. Yes. So one more question is there. What are the animal models for autoimmune disease?
It depends upon the type of the disorder. There are animal models. There are animal models for type 1 type of diabetes, animal models for rheumatoid arthritis, for psoriasis. There are animal models available.
Okay, so we can go through the literature only. That could be the
when we can get based upon the suppliers. Suppliers will be maintaining different types of animal models, and based upon our requirement, they will be providing that type of.
Okay, so one more questions. A few questions only I will take, sir. Which pharmacological experiment can do effectively in Indian environment?
All There is no variation in the Indian environment or any other environment. It is only the variation in the laboratory conditions. To what extent, to what effect, to what extent of their laboratory is effective. There is no such variation in whether it can be done in this country or in other country. The only thing is to what extent your laboratory is sophisticated in handling that sort of experiments.
Okay. Okay, sir. Thank you, sir. Last question, sir. I would like to take a rotarod test for evaluating motor functions in diabetic rat can be used.
It depends upon the intensity of disability or you can say neuropathic conditions that the diabetes has induced in that animal. If the extent of neuropathy or damage to nerves that this diabetic model has induced in that animal, then it is difficult, we cannot observe, or we can say we cannot get the results in the case of this rotarod test. If the intensity is more, the damage to the nerves it is more, automatically the ability to walk on this rod also will be difficult. So in such cases, they should be following some other models such as paw tracking model, where the paw will be dipped in some marker or some ink, and the extent to which the impression is created on a paper when it is walking, that can be taken into consideration. If it is a acute model, there won't be any difference in nerve damage, or in such cases motor activity will not be affected.
Okay. Okay. Thank you, sir. To what extent animal pharmacological models are reliable to start a phase 1 clinical trials in humans?
They are 100% reliable, and it is mandatory for us to clear this preclinical studies before we go to any human trials such as phase 1 trial. And all these preclinical studies, apart from in vitro, all these in vivo studies also have to be satisfactorily completed, and they should be meeting the required standards, so that then only it will be accepted for phase 1 trials.
Thank you very much, sir. Thank you very much for answering the question. And we are getting so many good remarks regarding the session. Thank you for giving the detailed information about the validity of the experimental models and the models for the behavioral pharmacological studies. Now I request our principal, Dr. V. Alagarsamy, sir, to give to present a token of appreciation. Principal, sir.
Yes, madam.
Yes, sir. So please, over to you, sir.
So thank you, Dr. Jitendra, for your wonderful lecture. Hope all our audience must have enjoyed. And there are so many questions we could observe in the net. So that shows how much they understood your lecture. So really it was a good lecture and everyone enjoyed the session. So as a part of token of our appreciation for your valuable time that you spent with us, kindly accept this certificate and the memento, sir. Kindly accept it, and thanks on behalf of all the participants, management, and faculty members. Kindly accept this memento, sir.
Thank you, sir. Thank you very much.
Okay, sir. Okay. Thank you very much. Thank you, one and all, for joining us this national webinar, and hope all must have received. Even if you have any more queries also, you can put up. So Dr. Jitendra will address to you. So you can, all the questions we can raise to us. So definitely he will come back with a good addressing. So with these few words, I thank one and all. Over to Dr. Vishaka.
Thank you very much, sir. Thank you very much. The feedback link is shared shared on the screen and sent through WhatsApp group. Kindly share the feedback, and we will receive the certificate for the participating in the conference. Thank you very much. Thank you, sir. Thank you, Jitendra, sir. Thank you, Principal, sir. Shall we?
Thank you, sir.
Shall we
Thank you,
conclude
Dr.
the session,
Saranand.
sir? Dr. Jitendra, sir.
Yes, sir.
Yes, sir.
Thank you.
Thank you, sir. Thank you.
Thank you, sir. Thank you very much, sir.
Dr. Saranand, we can sign off.
Okay, sir.
Okay.